Hello friends, welcome back to NitVed Bioscience. Let's continue our discussion on structure of the protein. Protein, we all know that the synthesis of protein, it is a continuous step and it is formed by the process known as a translation. So through translations, we will get the polypeptide chain where the monomer is amino acid. Means all the amino acids, they are linked together by a bond. That bond is known as a peptide bond, and this structure is known as a primary structure of the protein. We also know the many examples of proteins like immunoglobulins, enzymes, hormones, cell receptors. So these are the various examples of the proteins. So what is the difference between them and these structures? So this is the primary structure, but it is not functional protein. Okay, this is just a polypeptide chain and means long chain of amino acid, but it is not the functional. So to make the functional protein, it has to undergo the various levels of the modifications, that is either the folding and some chemical modification. Okay, so apart from chemical modification, folding is very important for the function of protein. There are various levels of the protein folding. These are primary, secondary, tertiary and quaternary. Okay, primary level, secondary level, tertiary levels and the quaternary levels. Primary levels means the long chain of amino acids, bond involved, and phi and psi means angle formed through which the bending takes place. All these we have discussed in the last class. And also we had discussed the Ramachandran plot, so you can watch that video. Link is available in the description box. Today we will continue our discussions and we will see the other level of folding. So secondary, tertiary and the quaternary. Tertiary and the quaternary, these two levels, these are actually responsible for the functionally active protein, but first it has to undergo the primary, secondary, tertiary and the quaternary. So these two we will study in the next class. Today we will have the focus on the secondary structure of the protein. How the secondary structure is formed. So this primary structure of the protein means the long polypeptide chain. So if in a long polypeptide chain, some local or the small portion, okay, please understand carefully, long chain of amino acid, if the small portion of that amino acid chain, it undergoes into the local and the repetitive level of folding, some repetitive like either helical or zigzag pattern. Okay, some few amino acid will undergo this type of the folding or the modification. So then it is known as a secondary structure. There are various examples of the secondary structures. So what I am trying to say that the secondary structure is not the whole polypeptide chain will undergo into the modification. Some the small or few amino acid and repeatedly form a structure. So either helical or zigzag structure. So that will become the secondary structure. So there are various secondary structures like helical structure, just we had seen. It is a spring-like structure. Okay, coiled or the spring-like structure of the polypeptide chain. So that is a helical structure. The most common example of that is the alpha helix. And is the first is the alpha helix. Other, if the polypeptide chain are present in a zigzag form and they form a sheet, and these are the more than one polypeptide chain, then the other example is the beta pleated sheet. Okay, helical structure, zigzag structure or beta pleated sheet, then beta bends, collagen triple helix, collagen triple helix, non-repetitive structure, and super secondary structure. Okay, so these are the some example of secondary structure: alpha helix, beta pleated sheet, beta bands, collagen triple helix, non-repetitive structures and the super secondary structures, out of which the most common are the alpha helix and the beta pleated sheet. So this we will cover in the next class. In the today, first we will elaborate the alpha helix structures, or we will see the alpha helix structure in detail. Helical structure. Okay, secondary structure means alpha helix, it's a helical structure. So before directly starting the alpha helix, first we will see how this helical structure is formed. So this helical structure is formed by a bond, that is a hydrogen bond. So we can say the secondary structure is formed as well as stabilized by hydrogen bonding. So how this hydrogen bond will form, and which is responsible for the secondary structure of the proteins, because this hydrogen bond is present in the alpha helix as well as in the beta sheet as well. So first we will understand the hydrogen bond. See, just we had seen that this is the primary structure of the proteins, where the amino acid are present and they are attached to each other by the covalent bond. So if we number this amino acid, so the first one will become the N, then N plus 1, N plus 2, N plus 3, N plus 4 and so on. Okay, N plus 5. So how, let's see how hydrogen bond is formed. We know that this is a polypeptide chain, this is a peptide bond. Okay, this much CONH is a peptide bond. So here in the peptide bond, this oxygen is more electronegative and the hydrogen is electropositive, partially electronegative, partially electropositive. So the hydrogen bond is formed between partially negative oxygen from one amino acid with partially positive hydrogen from another amino acid, which is few amino acid away from the first one. Okay, so that will form the stable secondary structure. So suppose this oxygen will form a bond with the hydrogen. So this will come closer to this. Okay, this chain will come close here and it will form a bond. Again it will form bond with another hydrogen. Okay, so that way ultimately it will form a like a coil like structure. Okay, so means the oxygen from the first amino acid, it can form a bond with the hydrogen from second amino acid or from the third amino acid. Okay, so if these things will happen repeatedly, so it will form a coil like structure where the hydrogen bond are present like this. So there is a possibility that the first oxygen from the first, it can form bond with the second, third, fourth, but the most stable structure is the one in which there is a bond formation between the first, means nth position, and n plus fourth position, means this hydrogen bonding. So oxygen from the first amino acid, we can call it as a N, and the hydrogen from the N plus 4 means first it will not form bond with the first, not with the second, not with the third and the fourth one. Okay, so N plus 4, when there is a hydrogen bond formation between the oxygen and the hydrogen, the number of amino acid means the first and the fourth, so that will form the most stable structure, and that is the alpha helix. So it doesn't mean that the other structures are not present. Other structures are also present, but that is a less common structure. So just we had discussed that secondary structure is formed by the hydrogen bond, and hydrogen bond is formed by the in between the carbonyl oxygen and amide hydrogen from the few amino acid away. So there can be the, if we collaborate it in one chart, so if we consider the first amino acid, so if we name the first amino acid by the N, so it can form the bond with the N plus 2, means first one is the N, then it will not form bond with the N plus 1 but with N plus 2. So if that secondary structure is formed, it is known as a pi helix, where the number of amino acid present in a one turn will be 2.2 and the number of atom will be 7. Likewise, if the first amino acid is N, and it will form a bond with the hydrogen, it will form a hydrogen bond with the N plus 3, not with the N plus 1, not with N plus 2, but it will form a bond with N plus 3. That is known as a beta helix. Amino acids in one turn of helix, one single turn of helix. So total number of amino acid present will be 3 and total atoms present will be 10. N and N plus 4 means first will be N and not with the N plus 1, not with N plus 2, not with N plus 3. So hydrogen bond will be formed between the N and N plus 4, that is known as the alpha helix. It is the most common and the most important secondary structure. Total number of the total number of amino acid present in a one turn of alpha helix will be 3.6 and the total number of atom will be 13. And one more structure, so that is N, there is a hydrogen bond formation between N and plus 5, that is known as a pi helix, and 4.4 amino acid present in a one turn and total number of atom will be 16. Okay, so this is the most common secondary structure or the helical structure, most common secondary structure of helix, that is the alpha helix. Okay, so I hope you are clear. So this way the hydrogen bond formation, if this is a N and N plus 4, so hydrogen bond, then the next one will form with the bond with another one. So likewise all the amino acids are interlinked with each other with the hydrogen bond, and hydrogen bond will stabilize the secondary structures. Hydrogen bonding, it is a dipole-dipole interaction. It is stronger as compared to the other dipole-dipole interaction, but weaker than the covalent bond, ionic bond, that we will know, but more stronger than the other dipole-dipole interactions, therefore it will stabilize the structure. Okay, so let's see the alpha helix in detail. This hydrogen bond which is present in the alpha helix or the helical structure, it is formed within the same polypeptide chain, means it is an intra-chain hydrogen bond. This is the helical structure and hydrogen bond formation will take place within the same polypeptide chain. So here the hydrogen bond form is the intra-chain hydrogen bond which is parallel to the axis of the helix. So if this is the direction of the helix or the axis of the helix, so hydrogen bond is parallel. So these are the two important things. Here the hydrogen bond is parallel to the helix and it is a intra-chain, means within the same polypeptide. Why it is important? Because this will make a difference from the beta plated sheet, where the hydrogen bond is perpendicular to the axis of the polypeptide chain. Okay, first it is a perpendicular, and second it is there we will see that there the hydrogen bond is the interchain, means there the hydrogen bond will form between the two different polypeptide chain in beta helix, but in the alpha helix hydrogen bond formation will take place within the same polypeptide chain. Okay, now so alpha helix, these are of two types, left-handed alpha helix and the right-handed alpha helix. So let's understand what is right-handed and the left-handed alpha helix. So for that, let's make a fist of your both hands where the thumb is stretching outwards. So thumb is the direction, means upward direction if we considering the rotation of the polypeptide chain. So it will move in this direction and this curled finger will demonstrate the rotation of the polypeptide chain. So if there this way, okay, so if this way the polypeptide chain will be curled, so it is known as a left-handed alpha helix, and if the polypeptide chain will curl in this direction and if it will move upwards, so it will be the right-handed alpha helix. So out of these two, right-handed alpha helix, it is the most common secondary structure which is found in the biological form of the proteins, right-handed alpha helix. So now let us understand all the dimension of the alpha helix. Suppose this is a one coiled like structure. We had discussed that the stable structure is the one in which the hydrogen bond formation will take place between the N and N plus 4. Okay, so here the number of amino acid present in the one turn of the helix will be the 3.6 amino acid. Okay, 3.6 amino acid present in a per turn. Then the pitch. What is the pitch? Pitch is the distance between the two turn of the helix. This is pitch. Pitch is the distance between the two turn, and that depends upon the hydrogen bond formation takes place between the which number of amino acid we had discussed. There can be the n and n plus 2, n plus 3 or n plus 5. Okay, this type of the hydrogen bonding also takes place, but here the structure will become either the stretched one or the compressed one as compared to n plus 4. So if the stretched one structure is a little bit the stretched one, sorry, here, then the pitch will be the higher, and if the compressed one, so here the pitch will be the less, okay, as compared to this n plus 4. So pitch here will be the 5.4 angstrom. 4 angstrom. So two things we had discussed: number of amino acid present in a per turn, second is the pitch. Now we will see the rise per turn. Rise per turn means the distance between the two amino acid. So pitch is a 5.4 angstrom. Total number of amino acid present are the 3.6. How 3.6? 3 complete and the partial involvement from the first and the fourth one. This much amino acid are present in a one turn, partially from the first, partially from the fourth, and the 3 complete. Therefore, the 3.6 amino acid are present in a per turn. Therefore, the rise per residue will be 1.5 angstrom. That is the distance between the two amino acid. Okay. So 3.6 amino acid present in a 1 turn. So if there are the 36 amino acid, it will form a 10 turn. Okay. So these helical structures, it is a rod-like structure which is stabilized by the hydrogen bonding. Hydrogen bond formation takes place. It is an intra-chain hydrogen bond which is parallel to the axis of the helix. Okay. So this way hydrogen bond, suppose. So it is always parallel to the axis. Okay, two structures, left-handed and the right-handed. So the most common one is the right-handed alpha helix. Hydrogen bond, where the most common is the n plus 4 bonding will takes place. Now we will see the examples where this alpha helix or the helical structure is a more common, and which amino acid it is most, which amino acid means favors the structures and which will restrict the formation of the secondary structure. So let us see the example. In alpha helix, the value of phi and psi will be, so phi will be minus 60 degree, psi will be minus 45 to minus 50 degree. Example of the protein where the this helical structure is most common, it is a myoglobin, troponin C, alpha keratin, which is found in the hair, nails and the skin. Okay. Now let's see the amino acids which favors the alpha helix structure. So these are the some example of the amino acids like methionine, alanine, leucine, glutamate and the lysine. This amino acid which favors the structure of the alpha helix. But there are some amino acid which is also known as a helix breakers. These are the proline and the glycine. Okay. So proline, as there is a ring like structures, so where the nitrogen or the hydrogen which is present on the nitrogen, it is not available for the hydrogen bond formation. Therefore there will be the sharp bend will takes place while forming the helix structure. Okay. So proline, either it will present in a terminal, N terminus or the C terminus, but in the helical structures in the middle it will not present, it will form the sharp bend or it will destabilize the helix structure. Therefore, it is also known as a helix breaker. What happens in the glycine? So, in the glycine, the side chain only the hydrogen is present. Okay, so because of presence of only the hydrogen, it can have the wide range of the phi and the psi value. So that will also destabilize the helix structure. Therefore, these two, proline and the glycine, is known as a helix breaker, and this amino acid, it favors the helix formation. Okay, so I hope everything is clear about the alpha helix. In the next class, we will discuss the beta pleated sheet. Thank you.